Morning, everyone. Thanks for joining us today. I really appreciate your time and appreciate your attendance here. I am the new plant physiologist at PP Systems. I'm going to talk a little bit about my background, but today I'm going to first just tell you what we're going to be talking about today. I'm going to be talking about field assessments of A versus CI curves at a local Massachusetts farm and talk about some of the insights I gained there, looking at differences in species and cultivar responses to far red light, which is a very popular topic these days. So just a little bit of background about me. I started my higher education at the University of Akron in Akron, Ohio. There I got a degree in biology with a minor in chemistry. While I was there, I started working on research problems in that setting. I initially wanted to go into pre-med, but fell in love with the research aspect of science. The first thing I looked at when I was there was actually microbiology, quite a bit of a departure from what we're talking about today, but it is interesting because this was my first foray into fluorescence. I looked at single cell fluorescence imaging using confocal microscopy for a number of different applications for single cell respiration. But while I was there, I started to get an education in plant science. I spent a lot of time at the University of Akron Field Station with Dr. Randy Mitchell, who is still a biology professor there to this day and well-known ecologist. I looked at everything from pollinator foraging and native plant species and lots of wildflowers to actually imaging and extracting pollen from various types of flowers so that we can see what the structures of those different pollen granules look like. While I was also at Akron, I started working at a startup started by a few of my classmates. They decided to build a vertical farm inside of a business incubator in Akron, and it is still running to this day. It's called Vigio Gardens. It is primarily hydroponic type of production style in vertical racks like you see here. The purple lights are a dead giveaway, but they moved a lot of produce through that place. I helped build some of these structures and started really working on actual research problems while I was there when we have to try and manage a startup farm and the various problems that might occur in agricultural capacities. From there, I really started to get this interest in controlled environments. And so I moved on to Ohio State to work with Cherry Kubota at the Controlled Environment Plant Physiology and Technology Lab. And I worked on a couple of different projects while I was there. I got my master's working on grafted watermelons and abiotic stress tolerance of watermelon in the seedling stage. This is a very applied problem that only really applies to that applies mostly to the grafting industry, the commercial grafting industry that occurs there, but we're merging two types of plants together to create a stronger transplant. And once I completed my master's in this, I stayed on board with Dr. Kubota to work. Sorry, a picture of me and a couple of lab members at Ohio State, but I stayed on to work on my PhD in vertical farming type of applications. I worked on lettuce tip burn, which is a physiological disorder due to a nutrient deficiency of calcium that occurs in lettuce, typically inside of this vertical farm setting. So I spent a few years there and wrapped up at Ohio State this past spring and then joined the PP Systems group in July. So now that I'm at PP Systems, the sort of aspects of my role here are rather new for me, but the main goal is to start building somewhat of an in-house research capacity so that we can do some of this science firsthand and we can start to advance, we can put in more effort advancing the field of physiology and our understanding of plants and their responses to their environment. Also, my role is to provide technical and research support to folks like you attending this meeting. So please feel free to reach out to me whenever you have questions or need assistance in your experiments and using the type of equipment that we produce. And I'm happy to talk to you about any and all of your research goals. I'm also here to provide training and education about plant physiology and how to use our instruments. There's a lot that can go into that. And so in various applications of various different approaches. So I'm here for that type of support. And also we want to increase our ability to reach out to the greater academic community and the commercial communities and collaborate a bit more to advance plant science for everyone. This is obviously an important goal to move the future of agriculture forward, and also many other types of environmental sciences. And so I'm here to try and build a bridge to support each other in this greater community. So moving on to the topic of the day, I investigated some physiological mechanisms at a local Massachusetts farm using our equipment, primarily the Cyrus 4, with a chlorophyll fluorescence module and light unit attached to our leaf cuvette. Fortunately, the local farm near us, a U-pick operation, is very, very excited about having us come on board and join their efforts. And we are able to go and sample many different species of plants at our convenience, which is an amazing resource to have in this community. And so today we're going to be talking about a couple of different things. The methodology I used generally here is A versus CI curves. A versus CI curves are a way of estimating the photosynthetic efficiency and capacity of various plant species at the leaf level. You can see here the common sort of common trend for C3 and C4 plants, C3 in blue and C4 in red. On the y-axis, we're going to be looking at assimilation, and on the x-axis, we're looking at the intercellular CO2 concentration. The process for this is essentially increasing the amount of CO2 going into the leaf cuvette. We can measure then the consumption rate of that carbon, which allows us to translate it into a value for assimilation of carbon. And then intercellular CO2 is simply the CO2 concentration inside of the leaf. So basically, the total concentration minus the consumption rate of carbon is what gives us an estimation of intercellular CO2, or as I'll call it, CI, throughout this presentation. And there are pretty stark differences between how C3 and C4 plants assimilate carbon from the atmosphere. We'll talk a little bit more about that, but A versus CI generally is a way to investigate the plant's health, any stressors that might be impacting the photosynthetic ability of these plants, as well as damage to the plants. It's an important thing to look at for climate change research as CO2 continues becoming more abundant in our atmosphere. We want to understand how plants are going to respond and how productive they'll be in the context of various climate scenarios. We also may want to compare different plant varieties to look for differences between different types of plants. This is also a common approach when we're screening different lines of plants through a breeding approach. Basically, if we wanted to create a high-yielding plant, we might screen across various different lines for highly efficient photosynthetic high photosynthetic efficiency, and the ability of those plants to withstand the various stressors that might reduce that efficiency. Another aspect of this research and what we're going to get into is far red. Now, far red is a series of, is it basically as radiation increases above 700 nanometers, which is the end of the visible range of red. We do have some photons that are invisible to us, but visible to plants. And it has been found that plants can photosynthesize with these far red photons. But far red also plays several other roles in the plant's production. So we can see here, this is our visible range in the gray box with our wavelength on the x-axis. And here is where far red falls. Now, far red can be up to 19% of the typical sunlight spectrum. And by 19%, I mean that the sum of this gray box with the red outline can be up to 19% of the sum total of this lighter gray box with no outline to the left. So there's quite a lot of photons coming from far red directly from the sun that plants are using in ways we did not formerly understand very well. So as I said, 19% of these photons, and we have a higher fraction of this far red beneath plant canopies, which I'll talk about in a little bit. But there are many different responses to this far red light besides just driving photosynthesis. It can include phytochrome regulation, different shade avoidance responses, where we think about stem elongation or leaf expansion, and photomorphogenesis, basically affecting that morphology of the plant when exposed to far red light. Like far red light is very important at the end of day for many fruit-bearing species to induce flower initiation. So what we wanted to investigate here was this role of far red a little bit more. So besides the shade avoidance and morphology type of influence, there's also good evidence that the inclusion of far red in the light spectrum can reduce non-photochemical quenching through a certain pathway that all plants share. It also can regulate the growth of that plant via that phytochrome sensing response and can drive photosynthesis. But the caveat here is that it only really drives photosynthesis when in combination with our typical definition of photosynthetic active radiation, which is this visible light range. So far red needs to be in combination with this visible light to efficiently drive photosynthesis. And we also see in many species that there's an increase in photosynthetic efficiency when far red light is present due to driving of that photosystem 1. We commonly think of photosystem 2 as the primary driver of photosynthesis, but excitation of photosystem 1 is a major component, and far red is very adept at driving photosystem 1 to be more productive and efficient. So here's a picture I took on a walking path near my house. On the right side, we're seeing unfiltered sunlight come in through a gap in this tree canopy. And on the left, we're seeing shaded light coming through layers of leaves in this canopy. So when we don't have a filter on sunlight, we have this very broad spectrum of high intensity light, ranging all the way from the UV range through the far red up into the infrared range. And we see some differences here when we apply sort of this canopy filter, I'll call it, of biomass growing above a floor level type of species. So the plants over here on the left side in the shade are experiencing a much different light environment than plants on the right side in direct sunlight. So when we measure the light across these different wavelengths on the left side in the shade, we can see that far red penetrates deep into the canopy while many other wavelengths of light are sort of lost in that canopy. They're either being used in photosynthesis and above in the canopy above, or they're scattered throughout the lower canopy at a lower density. But we see a lot of that far red penetrating down to the forest floor, where there are often many plants growing along the forest floor that are primarily using this far red light in their photosynthetic reactions compared to when we have unfiltered direct sunlight. And you see some very different responses here. But we want to kind of think about this more deeply in a way where we can understand the role of far red in these types of environments. So some of the challenges of far red, just to outline before I get into the data, we don't really have a good model for photosynthesis in either cultivated or non-cultivated plants that include far red as one of the major inputs to those models. We've almost ignored it to an extent. And so we've made many models over the years of both cultivated and non-cultivated plants in agricultural or horticultural or simply ecological settings. And none of those models are perfectly fit, but we can improve the fitness of these models by including some better understanding of this far red and its influence on the plants. In many horticultural settings, there's now a question of whether we should apply far red light, especially in controlled environments and things of that nature, where we're crafting LEDs to fit in those environments and provide different light spectrum to plants. The question is now to include or not to include far red. We also want to better understand some of the morphological responses and the ability crafting the ability to crop steer plants by using far red light and exactly what that inclusion of far red will do to change the morphology of those plants. And what we really don't have a great grasp on yet is the variation of response to far red among many plant species, especially when we think about photosynthesis and how efficiently far red may or may not drive photosynthesis in combination with these other photosynthetically active photons. So some of the methodology I used to determine the far red and ACI data that I collected, we created PP Systems created a fast ramping mechanism, basically to better understand and quickly collect data to generate ACI curves. So in about 5 minutes or so, we can ramp CO2 from a very low level, in this case, about 100 micromoles per mole or ppm up to 1,000 micromoles per mole. And in those 5 minutes, at a data collection rate of 1 record per second, we can create a very, very nice ACI curve that gives us a lot of insight about the plant's response to the plant's photosynthetic capacity across a wide range of CO2. You can vary many other factors here, including temperature, and, humidity, and various other things at the level of the cuvette. But we developed this process to quickly collect these curves as opposed to traditional methodologies. And the simple way we did that was modifying the gas lines on OSIRIS-4 to be a slightly more adjustable in volume. This reduces any delays between the two infrared gas analyzers so that you can get accurate, precise live readings of photosynthesis and CI across a range of CO2 in a very short period of time. We call this the SCORE method. So I won't go too much into the methodology behind that, but basically we wanted to be able to quickly collect this type of data. ACI curves in general look, here's just a comparison between the steady state, the traditional type of collection. For every red X, we set a CO2 concentration. We clamp that cuvette onto a leaf. We allow the leaf to get to a steady state. And then that takes about 2 to 4 minutes, depending on your species. And then we'll increase the CO2 to the next X point and allow the leaf to reach steady state again. And so for these 8 points, it took about 40 or 45 minutes to capture that data. On the same leaf, we also tested our SCORE method, where we're collecting data at 1 record per second. And in about 5 minutes, we can collect what used to take a researcher about 45 minutes to do. And the data agrees very strongly with between the two methodologies. And so we're very confident and proud of this, this new capability that we've built into OSIRIS-4. And from this type of data, you can also extract many different variables that are important to understand in the context of looking at assimilation and photosynthesis of plants. So things like the VC max, the J max, your TPU, RD, and GM are all important physiological parameters that help us understand the efficiency of various different components of photosynthesis, including the electron transport chain speed and the efficiency of photosystem 2. And so this is a very useful tool where we can extract lots of extra data from this type of approach that's very valuable for physiologists to look at. And to look at the different different effects of far red, we have installed far red LEDs into our leaf cuvette in our light unit. And we can achieve up to 30% far red intensity relative to the PAR intensity. So if our PAR intensity is about 1,500 micromoles, then we can achieve 300 and 30% of 1,500. And so allowing us to use this adjustable light spectrum is very easy to do and important for us to be able to rapidly take this kind of measurement. So throughout this presentation, we kept that far red intensity. We changed the far red intensity, but we kept the total photon intensity the same at 1,000 micromoles. And I'll tell you a little bit about that in a moment. So like I mentioned, we went to our local farm, Cider Hill Farm in Amesbury, Massachusetts, and they have a variety of crop species we were able to test. We modeled this photosynthesis using the A versus CI curves for several species and cultivars. We ramped CO2 upward from 200 to 1,000 micromoles per mole at a rate of 150 micromoles per mole per minute. So in about 5 minutes, 5.5 minutes, we're collecting a complete ACI curve. For corn as a C4 species, being that the asymptotic maximum of photosynthesis is quite low on the CO2 concentration side, we only ran that one from 0 to 300 micromoles per mole of CO2. We then identified some differences in photosynthesis with differing fractions of far red light. So we replaced the photons with 0, 15, or 30% far red intensity. So while the total light intensity was 1,000 micromoles, at 15% far red, we would only provide 850 micromoles of photosynthetically active radiation and then 150 micromoles of far red. The spectra of the photosynthetically active radiation was 1 to 1 to 1, blue to green to red, which is a pretty broad spectrum white, kind of modeling what sunlight puts out. And this was our approach. So we ran 3 different ACI curves per cultivar or per species at 0, 15, and 30% of the far red. Some hypotheses we had going into this was that the maximum assimilation values between species will be quite different. There are some species that are simply more productive than others on a per varia basis, but the different species will have similar shapes, sort of, a linear initial response and then an asymptotic maximum photosynthetic rate that is pretty typical of the figures we've already seen. I also know that due to the anthocyanins and other photoproductive pigments in red leaves that we would see in red leaf cultivars or red leaf type species, that there would likely be a lower maximum photosynthetic rate than green leaf type cultivars. So if there's a green and a red leaf lettuce, my assumption was that the red leaf lettuce would be less productive on a per square meter leaf area basis. Also, due to the recent research into far red and the added efficiency of driving photosystem 1, my hypothesis was that the increasing proportion, meaning from 0 to 30% as we keep going up, we will see a faster photosynthetic rate, or higher photosynthetic rate, as we increase the amount of far red in the light spectrum. But that maximum benefit might be achieved somewhere between 15 and 30% far red, seeing as sunlight contributes a maximum of about 19% far red light across that total photon range. So my assumption was that there would be some kind of limitation there between 15 and 30% far red, where we'd see probably a little bit less photosynthesis at 30% than at 15. So we examined a handful of species. We looked at 3 different cultivars of apples, 2 cultivars of Swiss chard, which you can see in the rows in the middle on the bottom, green and red leaf type Swiss chard. Lettuce, 2 cultivars, green and red leaf lettuce is on the very right-hand side, a little bit blurry, but in the photo below. We also looked at 1 cultivar of strawberries and 1 cultivar of corn. So this process looked a bit like this. We would clamp onto leaves of that given cultivar or species. We would then ramp our CO2 from 200 micromoles up to 1,000 micromoles once we achieve the initial steady state. Then I would go ahead and, once that curve is complete, change the light spectrum a bit, increase from 0 to 15% far red and then 15 to 30% far red, while keeping that total light intensity the same. And then I'll run this curve again. So basically a very quick high throughput process, running multiple ACI curves on the same plants, and then looking at these results, which we'll do together in a moment. So I took a total of 27 ACI curves for those 5 total species. This data collection took around a total of 5 hours, including the setup, including moving around the farm to various locations to seek out the plants that I was interested in, including the breaks in between plants so that I could make sure I set everything up appropriately, periodically checking the data. But I collected over 9,000 total data points relevant to this work in that 5-hour period. And I could have probably gone faster if I had chosen to, but at a leisurely pace, about 5 hours to collect a huge data set was great. And I hope to continue doing that and sharing this with you all. So now we're going to get into some of these results, and I'll just go by species down this list, and then we'll wrap it up with a few conclusions here. But when we look at green versus red leaf type lettuce, green on the top and red on the bottom, again, we have our assimilation on the y-axis and our CI on the x-axis. For the green leaf lettuce, we see a higher photosynthetic rate, maxing out around 25 micromoles per meter squared per second. But on the lower figure, we see that the red leaf lettuce maxes out around 13 or so in that assimilation rate. So quite a big difference between green and red leaf lettuce, which is not unexpected in this case. So for the green leaf lettuce, however, so I'm sorry, I probably should say that the color coordination here. So along the bottom, you can see 0, 15, and 30% far red. The kind of grayish green in the back here that I'm pointing at is 0% far red. So that's just 1,000 micromoles of purely par light. And then this sort of tealish color is 15%, and the dark green is 30%. And you'll see that on each of the subsequent slides as well. The colors do not change. So with green leaf lettuce, we actually saw that about 30% far red light decreased along this curve, as we see here, and 0 and 15% far red light was very, very similar in this curve, almost identical. But the green light actually in the linear phase of this ACI curve was slightly less driving of photosynthesis than when we had less far red light. For the red leaf cultivars, there was similar photosynthetic rate across the entire curve, regardless of the far red percentage. So that's rather interesting to see the difference in just these two cultivars with, and this could be somewhat related to the anthocyanins and pigments, but it is interesting to see that there's quite a stark contrast between those two in the response to far red light within the same species. When we look at Swiss chard, we see some interesting results as well. When we have again, we see a similar thing where the green leaf Swiss chard had a higher photosynthetic capacity than the red leaf Swiss chard. We also saw that with increasing far red percentage, we do see an increase in photosynthetic rate. In green, it's pretty big difference. In red, it's actually just a small increase only in the linear portion of this ACI curve. And so, this could be related to things like anthocyanins and photoprotective pigments in red leaf Swiss chard, but it is interesting to see that the green leaf Swiss chard had a much higher photosynthetic rate across that entire range of CO2, which is not so similar to how the red leaf Swiss chard responded here. Now, corn is a bit odd in this figure. Obviously, we're reaching an asymptotic maximum a bit faster, only around 300 micromoles per mole of CO2. But we see that there's a higher photosynthetic rate with increasing far red after the initial slope. So they diverge there just over 60 or 65 micromoles per mole of CO2. And we see a consistently higher photosynthetic rate with far red light than without it across the rest of this figure. I don't have an excellent hypothesis yet as to why this is the case, but it is nonetheless an interesting result to see in this field-grown species. Now, with strawberry, we had a bit of a different response than the other species. We actually have a lower rate of photosynthesis with any amount of far red light. Regardless of the amount of far red light that was included, the photosynthetic rate was almost identical across this ACI curve for 30 and 15% far red. But with 0% far red, we see that there's a much higher photosynthetic drive across that entire range. Strawberry has been known to be very sensitive to far red light in terms of its morphology and its flower initiation, especially at the end of day. But it is a surprise to me that there's a lot of talk about including far red light and light spectrums for controlled environment grown strawberries, but it is very surprising to me to see quite a stark contrast here in the photosynthetic rate when far red is or is not included. And apple here might be the most confusing of all for me. We obviously expect various species differences, and maybe the leaf color of various cultivars would contribute to the differences in ACIs we see here. But for apple, the variation among cultivars was profound for me. We see either a decreased photosynthetic rate with far red light, an increased photosynthetic rate with far red light, or no change in the photosynthetic rate with far red light. So these three different cultivars were grown in rather close proximity to each other. As far as I know, there are no different management styles or treatments of these different apple trees, but we see a very big difference in how they respond to far red light and even their maximum photosynthetic capacity. So there's a lot of variation here, even within a species that is not fully explained, I think, in the literature. So just returning to kind of what these hypotheses looked like at the beginning, we thought that there'd be similar shapes between these A versus CI curves within species. That's somewhat true. There were some differences that were strong there, but we may just need more data to really see how exactly these different species are responding to measurements we took. We did find true that the red leaf cultivars are typically going to have a lower maximum photosynthetic rate than the green leaf cultivars within a species. We suspect, obviously, this is due to the anthocyanins and photoprotective pigments, but there could be other things happening here. But from the literature, this seems pretty typical for red leaf cultivars versus green leaf cultivars within a species. We also thought that increasing the proportion of far red photons will increase that photosynthetic rate, but that wasn't necessarily true. We saw increases, decreases, and no changes between different cultivars and species with the inclusion of far red light or the absence of far red light. So regardless of the intensity of far red in that spectrum, we saw quite different responses among different species, which merits some further research, I think. And we also thought that maximum benefit of far red would be between 15 and 30%, meaning that we would see maybe a lower photosynthetic rate at 30% than far red than at 15%. But we actually didn't see that. There was, I think that was true in one of the cases for one cultivar, but in most cases, that response was not as anticipated. We saw some plants with their maximum photosynthetic rate with 30% far red, but other plants with their lowest photosynthetic rate with 15 or 30% far red, any far red at all. So that doesn't exactly answer that question, but it definitely is something that I will continue looking into. And so just to kind of conclude some of these points here, far red light is always present in outdoor conditions, with that sunlight contributing about 19%. of those total photons in the far red range. However, there are sometimes drastic species and cultivar specific differences in the photosynthetic rate when exposed to far red light. Not just the presence or absence of far red, but also the intensity of far red in relation to the other photosynthetic photons. Far red does have many roles to play besides just driving photosynthesis. So the photomorphogenesis that we talked about, the stem elongation, leaf expansion, shade avoidance responses, regulation of the phytochromes. There's a lot of different aspects to how far red is impacting these plants, even flower initiation. So it's not purely something that we should look at through a photosynthetic lens, but this is nonetheless information we need to understand. And so kind of the next steps in this far red future, I think, would be continuing to catalog and identify these species and cultivar responses to far red, and hopefully eventually linking that to what's going on at a deeper level physiologically in the photosynthetic pathway to understand exactly what these photons, what the role of these photons are in the future of plant science. It also is something where I hope to keep cataloging and releasing data so that we can improve our understanding of photosynthesis at the level of the leaf and the canopy and the ecosystem, because that's going to have a major impact on how we model different crops and understand the productivity of different crops. And besides that implication in photosynthetic modeling, we should eventually come to a point where we can build these photosynthetic models in combination with sort of carbon cycles and climate change scenarios so that we can understand and better adapt to the future of our environment and the future of our agricultural production, which is, I think, highly important for us to have a really good grasp on and understand as we go into the future. So I just want to say thank you very much. I really appreciate you attending here today. I'm happy to take any questions from the chat, and in a moment, once we start going through the chat, we can go ahead and ask folks to unmute and ask questions as well. So if you would like to unmute and ask a question, I ask that you just throw something in the chat to ask for the ability to unmute today. But I really appreciate your time and attention. Thank you so much.
Thank you, John. There's actually quite a lot of questions in the chat.
All right.
If you want to take a look, or I'm happy to read them to you. Daniela Zakaria is asking, could you please specify what the percentage of far-red radiation relative to total incoming shortwave radiation is in an average sunny day?
Yeah, so I think there's actually a movement to kind of change this to considering like the far red fraction of the total light. I'll have to look in that paper exactly, but basically that sum of photons between 400 and 700 nanometers is usually, I'm sorry, the amount of far red is usually equivalent to about 19% on an average sunny day of the total sum between 400 and 700 nanometers. I see his follow-up question with orchard floor management affecting the amount of far red radiation being transferred or reflected. Yes, that is an area we need a lot more research to understand the transmittance and reflectance of far red in different horticultural contexts, especially bare ground versus cover cropped ground. And I haven't seen any really good papers examining that yet, but that's something we should definitely investigate further. And I'm not sure specifically in the context of orchard floor management.
Okay,
oh,
and then I'm sorry, go ahead. Did you want to just read down through?
Sure.
Okay,
Yeah, I can do that. So it looks like a question from Raul, which is what adaptation time do you use between each CI point? So in steady state applications of A versus CI curves, typically you use 3 to 4 minutes between each CI point. In this case, when we're using our score ramping process, there's 1 second between each data point because the reference and analysis ergas are reading at the same time. There's no delay between either of them. So that's helpful. I see a thumbs up there. Another question from Arul about VC max and J max. We do not calculate it with the equipment. We do fit it afterwards. We have several tools on the PP Systems website linking to those various tools. But if you want, you're welcome to send me an email, and I'm happy to include those tools myself, Raul. Thank you. Yes, well, the far red percentages are compared with, they're compared with a combination of red, green, and blue light. So 1-to-1-to-1 ratio of each of those. So if I have 850 micromoles of par photons, that's, a third blue, a third green, a third red, and then adding in 150 additional micromoles of far red. Nathan, thank you for your question. I'll just repeat it in the chat here. Nathan says that we focus on the effects of far red light on leaf morphology and photosynthesis, but what direct effects do you think far red could have on the fruit of crops like strawberries or tomatoes in CEA? That's a great question. The effect of far red on the actual fruits themselves is very interesting. I have really thought about this mostly through a leaf context, but you're igniting some interest here to maybe look at this through a fruit context. But I'm not, besides their effects on the plants, besides far red's effect on the plant's morphology and development, I'm not sure how it actually affects the fruit set and fruit development. So that's very curious. And then Daniel Zakaria asks if there's any far red sensors for diurnal continuous measurements on the market. I believe there are. If you want to send me an email, I think I can point you in the right direction for some of those. And at this point as well, it looks like I've gotten to the end of the chat questions. If anybody wants to ask a live question, feel free to put something in the chat or just raise your hand, which is on the in the reactions along the bottom.
So John, thank you for your excellent presentation. I wanted to ask you if the Cyrus tree can be adapted or modified for such measurements, or we have to scale up to the Cyrus 4. I don't think there's a lot of difference between the two, but we just have two Cyrus 3 in our lab. It would be difficult.
Sure. Yeah, so for the ACI application, definitely. I've tested out our square method for doing rapid ACI curves. That definitely can be scaled up. I'm actually in the process of writing an application note for that. So if you want, feel free to send me a message, and I'm happy to share that with you. As for far red, we haven't gone back and modified the Cyrus 3 light units. But definitely reach out to me. We'll talk more about how we can do more far red type of research as well, if that's your interest.
Okay, thank you so much.
For anyone that wants to reach out, I'm still a bit new at this role here, but I'd love to hear more from each of you about what kind of things you're working on, and if there's any way to start building more of that bridge between scientists so we can better advance our industries forward.
Okay, well, if anyone has questions after this that they think of, feel free to email me or John directly, and we'll get you an answer. This will be up on the website shortly, and anyone who has registered for this will receive a link to it. So thank you again, John. Thank you for all the great questions, and we'll see you next time.
All right. Thank you, everyone.
Thank you for attending the webinar. Contact us today to learn more about the Cyrus 4 portable photosynthesis system, the fourth generation portable powerhouse elevating the high-level field research experience worldwide.
worldwide.